All right, thank you everyone for coming. Just a few announcements before we get started. Tomorrow night is the Lincoln Science Night, 5 to 8 p.m. I think we sent out a link to register, so if you're interested, helping out, teach science, physics, some kids, sign up. Yep. Who came out last week to the aeronaut event? Who was there? All right, so those not on the council, what did you think of it? Just top of your head, like just roughly initial thoughts. It's good. Okay. Okay. Good. Okay. All right. We're gonna try to see if we can do more stuff like that in the future. So just good to get feedback. Yeah.
Are you gonna say something about the octopus?
Yes, that's on May 8th, right?
Right.
And so we have our next talk will be in May 8th at Opticos's in Wakefield. So we'll send out an email announcement as we get closer for that one as well. So want to introduce the speaker today. She's coming from Boston University. Name of the talk is Uncovering the Pathways of Excitation, Energy, Flow, and Light Harvesting Systems with ultra-broadband, harvesting systems with ultra-broadband 2D. Very looking forward to deciphering that title there. Yeah.
Thank you.
And this is Min Jung Sun from Boston University Department of Chemistry. Thank you very much for coming.
Thank you. Thank you for attending my talk and for having me here. It's a great pleasure to finally be outside of Boston proper, I guess. Wait, okay, great. All right, so I will be talking about two different stories today, which are mostly my past work because I'm still pretty new at BU, and so I'll talk about what my work is, what my group is doing currently at the very end of the talk. But mostly this talk will be about my past work during my PhD and postdoc, using a technique called ultra-broadband 2D electronic spectroscopy to decipher, like Sanjay mentioned, how energy flows in these light harvesting systems. And my computer froze. Okay, so I mean, I guess if you know academics who know people who are looking for labs, my lab is pretty new. We work on these types of things. I guess I can't use the pointer because it's reflective. But we work on ultrafast spectroscopy. We work on polaritonics. Let me turn on my fake laser pointer. Here we go. Yeah, and we work on nanomaterials, so I'll talk about some of that, polaritonics and ultrafast and nanomaterials in this talk. Right. And of course, this also stopped working. Okay, I'm sorry. This actually stopped working now, and so I don't really know. Okay, here we go. So here are the people who made all of this work happen that I'll talk about, which is my postdoc group, Professor Marty Zanni's group at Wisconsin-Madison when I was a postdoc. The second part of the story on carbon nanotube polaritonics will be is a beautiful collaboration between our Zanni group and my collaborator, material scientist Mike Arnold. And then the first part of the story on natural light harvesting system is the work that I did when I was with Gabriela Schlau-Cohen at MIT as a grad student. And these are the people that helped me do the experiment. And of course, I have to thank my collaborators, plant biologist in Verona and Pavia in Italy that provided the proteins that I studied in the first part of the story. And also a couple simulations that we did with Professor Stefan Cohen at Montreal that I unfortunately won't have time to talk about in detail. Okay. So here's my research career, or the theme of my research career, past and present. I'm really interested in thinking about systems that harness light energy from the sun. And so as we all know briefly, the workflow of these systems is that they capture the light energy from the sun like this, and then the energy has to transfer to some side of useful work or function, for example, charge separation. And as a physical chemist, and in particular an ultrafast spectroscopist, I'm really interested in looking at this transfer portion of the workflow, asking questions like how does excitation energy flow in these materials or systems, and can we control it based off of the understanding that we can gain from performing these ultrafast experiments using lasers. Okay, so basically, if I can understand the energy flow, I can maybe design some or come up with some design parameters to control the energy flow, and maybe we can inform to the industry people or like the materials engineers who are actually making devices. Here is how you should put together your building blocks for better performance. That's sort of the theme, overarching theme of my research career so far. And so today I'm going to focus on two different systems you see in these photos. One is a natural sort of light harvesting systems, photosynthetic systems we can find in nature all the time. And then the second half of the story is about synthetic nanomaterials, in particular carbon nanotubes, which I'm showing here, and how photonic actually modes can promote some really long range energy transfer that's not seen outside an optical cavity is going to be the topic of the second half of the story. And so I'll start with this story about the green plants, which is really focused on understanding the fundamental steps of energy flow in these leaves. So basically, the major question that I tried to answer during my PhD when I was doing dissertation work was, how do green plants actually regulate the flow of solar energy? If you think about these photosynthetic systems, they are super efficient at capturing the light energy from the sun, but they are also really good at regulating, controlling the balance between if there's too much light energy, for example, they can get sunburned. Like how do they actually prevent themselves from getting photo damage from too much light? And so those are the things that I am going to be talking about for the first half of my talk. So here I'm starting again with a leaf that I already showed you a couple times already, and they are a really efficient light harvester. And so if we zoom in onto the photosynthetic machinery of these plants, there are actually a bunch of proteins. And so here I am showing you a bunch of these blobs, green blobs and dark blobs that are all proteins. And then these proteins are membrane proteins, and so they're embedded within a lipid bilayer membrane, which is shown in these gray area, okay? And so what happens when they absorb the sunlight, is that the absorbed energy transfers obviously downhill through this network of proteins, which are called light harvesting complexes, which are the antenna protein that actually capture the sunlight, to this reaction center where charge separation and energy production happens. So this is what we learn in our high school biology class and general biology class in college. But what they didn't tell you in that class is that this only happens on a cloudy day. What happens on a sunny day is that there's too much sunlight simply for the plants to utilize, and so it's actually not safe. Like I briefly mentioned in passing before, they can get sunburned or they can get photo damage. And so to protect themselves from such photo damage, they actually developed a safety mechanism by hijacking some of the population from this pathway to dissipate as heat, which is collectively known as photoprotection. If you are more interested in the biological mechanisms behind photoprotection, these are some of the papers coming out of the Niyogi lab at UC Berkeley. So basically, if you think about the sort of the two arrows that I'm showing that are competing photophysical pathways, it's really important for us to understand the dynamic balance in response to weather, right? We have sunny days, we have rainy days. Boston is not very sunny. It can be cold in winter where there's sunlight, the level of sunlight is just super low. We're gearing up for summer, and so the sunlight intensity is going to go up. And so we really need to think about how these proteins actually control their photophysical mechanisms to protect themselves from photo damage, but at the same time utilize energy efficiently. And what's really interesting, if you think about it, is that field studies on native plants out in the field actually projects that optimization of this balance of the competing pathways could result in about 30% increase in crop yields, which is pretty significant. And if you think about how photosynthesis actually can be one of the ways to sort of tackle the ever-growing demand in global food and energy, this is a really exciting sort of result that you can think about. And so one of the problems in terms of trying to understand this balance is that the mechanisms of dissipation, the heat pathway, the gray curly arrow that I showed you, is really not well understood. And part of the problem is that we know, so here's what we know that happens from these field studies or EM, electron microscopy studies, which is that on sunny days under a lot of sunlight, these proteins can actually sort of form arrays, closely packed arrays. So if you compare this diagram versus this diagram, this is what happened under this is what happens under highlight. But what we don't know are a lot of these parameters in the molecular level, such as what are some of the changes in pigment orientation. These are proteins I'll show you later, but these are proteins that bind pigments, light harvesting pigments. So what are some changes in orientations of these pigments that are bound within this protein? Obviously, if you change the chemical structure or a physical structure, you will change the electronic structure of these things. So can we actually open up new photophysical pathways or timescales that might matter for optimization of dissipation in a sunny in a sunny condition? Part of the problem that we don't know a lot about these things is because previous studies have been performed mostly in far from native or complex environments. And so what I'm saying by far from native is that a lot of these proteins have been studied in isolated, super isolated condition within detergent micelles, which is super different from these lipid bilayer membrane that I'm showing you in this diagram. So for example, we don't really know, okay, is there a role that the lipid membrane that's surrounding these proteins in the native system is playing in dissipation? We can't know that because we have been doing these experiments in a totally different environment that lacks the lipid membrane. What about the protein-protein interaction? Like I said before, on a sunny day, these proteins actually sort of form assemblies, but if you are doing this experiment in an isolated environment, we don't have a neighboring protein, and so we can't study that mechanism. What about the size of the array? Obviously, there are going to be arrays that are smaller than others. And so what does that sort of parameter play? What role do these molecular parameters actually play in the dynamic balance in response to weather that I mentioned? Of course, people can do and have done in vivo measurements on whole leaves or large membrane chunks of this protein, which actually has all of these interactions and parameters in the native environment. But a huge problem with performing in vivo measurements with lasers is that they really obfuscate mechanistic details because we don't know where things are happening. If you have a bag of many molecules bound to many proteins, and in addition, lasers can actually induce artifacts when you are trying to measure such a large protein array with such a large number of molecules. And so these are some of the limitations that prevented people from actually answering these questions about the role of the lipid membrane, protein-protein interaction, and array size in the light harvesting and photoprotection of these proteins. And so what I did as a graduate student was to develop this really simplified, reduced near-native model membrane to put in these proteins. And so here I am showing again these green blobs, which are called light harvesting complex 2, or in short, LHCII. They are the major light harvesting complex in plants. These are actually the most abundant membrane protein found on Earth. They bind about 30% of all chlorophyll that we see in life, which is pretty abundant, pretty significant. And so what I did was to basically take this little portion still bound within my membrane and turn it into a discoidal sort of model membrane system, which you show on the cartoon on the left. And so what the structure is, you know, I still have my proteins of interest bound within this lipid bilayer, and then I'm wrapping it around this disc with this protein called belting protein, which basically controls the size and the stoichiometry of the proteins within the system. So this is called nanodisk. I didn't develop this platform myself. This is actually pioneered by the Sliger lab at the University of Illinois, Urbana-Champaign. But we took this approach and we inserted these protein for the first time to apply this to the studies of light harvesting mechanisms in plant proteins. And so in this mechanism or in this platform, we can actually study all of these questions in the absence of complexity coming from the in vivo environment, and we're not putting them in detergent micelles, so we actually have a near native environment in a controllable manner. And so here I'm showing you some spectroscopy data, or actually not spectroscopy, this is microscopy. So this is TEM data where I am inserting by stoichiometry design on one protein, 2 proteins, 3 proteins, and 4 proteins within a per nanodisk. And these little white blobs that you see in each image are actually individual nanodisks, and the scale bar is about 100 nanometers. And so I can also analyze the distribution of the diameter of these objects. And so here you see that actually the mean diameter, which I'm plotting as delta D bar, is increasing as I actually put in more proteins, which actually tells me that the array size is growing, and perhaps the extent of protein-protein interaction between neighboring members in the array is also increasing. And so this is a data that I took on actually a laser which measures the fluorescence lifetime. These proteins are fluorescent, so we can read out the excited state lifetime using a time resolved fluorescence spectroscopy. And you can see that, yeah.
Is it disruptive to the photosynthetic pathways to have the fluorescence, given that it's a photo response?
It's
pathway?
one of the competing pathways. I'll talk about it in a second. Yeah. Yeah, so what these plots show is that you are seeing the wavelength axis, which is the wavelength of the fluorescence getting emitted from the protein, and then the y-axis show you the time scale. And so you can see by looking at these contour plots, as we increase the protein-protein interaction or the array size, we actually get fluorescence quenching of these protein assemblies. And if you think about fluorescence being another competing pathway, so before I didn't show you this pathway. So this is photochemistry of the photosynthesis that we call light harvesting collectively. This is a photoprotection, dissipation, and then there's this other pathway, which is this fluorescence pathway. Fluorescence quenching actually reports sort of on the level of dissipation. So that's basically why we use the fluorescence intensity or lifetime as a readout for the amplitude of dissipation. Of course, this experiment is really exciting, but it doesn't report on ultrafast timescale. This is at best nanosecond order. And so we do need an ultrafast probe to resolve the rapid component that precedes this quenching that we're seeing. And so that's when I turned to this ultrafast technique called 2D electronic spectroscopy. I don't know how many people in this room actually know a lot about this, but it's an ultrafast technique that is measuring data in 2 dimensions. That's why it's literally called 2D electronic spectroscopy in the visible and near IR. The way it works is I have my sample cuvette. So this is going to contain my nanodisk. I come in with a pair of pump pulses. So if you know about pump probe spectroscopy, this is a 2D version of that. And so these pair of pump pulses come in at a time delay. I'm going to call that tau. And so they prepare collectively an excited state population of your sample. And then I give it another time delay, which I'm going to call large T. And then my probe pulse comes in, which is my third pulse that monitors the relaxation of the excited state population prepared by the pump pulses. And then I have another pulse that comes in within this, we call this the boxcar geometry, just because there are 4 beams sort of like focusing onto your cuvette like this. This local oscillator is sort of a reference pulse that we interfere with the emitted signal like this because it co-propagates with the signal, the wave vector of the signal emitted. And so we can take the interference between the signal and the LO, and then we put our detector here. And then we Fourier transform, because we're taking a lot of the data in the time domain to retrieve these 2 axes, which is the energy of the pump or wavelength and the probe energy of the system. And so we can do this experiment by varying this large T time, which is the population time. So for example, if I do this experiment T equals 0 femtosecond, when all of your pulses are coming in at the same time, I will see something like this. You see these red blobs, positive blobs on the diagonal. And then there's this blue blob, which is negative signal that's corresponds to the excited state absorption. So these red blobs actually originate from the ground state bleach and the stimulated emission from your excited state. Now, diagonal, like I just mentioned, there are a couple diagonal peaks that are already always showing up there, and those illustrate the energy levels of the system that I'm monitoring. So let's consider really simple, this is fake data, obviously, really simple donor acceptor 2-level system. So here I'm gonna call this peak a donor peak. Here I'm gonna call this peak an acceptor peak. So we do see these diagonal peaks corresponding to the energy of the donor and the acceptor. What's the beautiful about these 2D experiments is that if I now vary the time delay like this, so I'm doing the experiment at 100 femtosecond, 500 femtosecond, you see that there is this off-diagonal peak growing in gradually as I increase the time delay. So this is kind of the optical version of 2D NMR, if you've done 2D NMR in your undergrad, I guess. And so we can monitor the intensity of the cross peak or off diagonal peak growing in, which I'm labeling with these purple boxes. And so I can basically look at the kinetics of that growth like this. And then that usually correlates whenever there's energy transfer from the donor population decays with a corresponding time scale. And so if I now fit these data to extract the time scale of the energy transfer, that's how I know that there is energy transferring out of these donor diagonal peaks into the acceptor state. And so these off-diagonal peaks are going to be the key peaks that I will be mentioning again and again in this talk. Okay, so here's again my pulse sequence for this experiment. One of the technical challenges in the field when I was a grad student, which is almost a decade ago, was that they have been using limited spectral bandwidth, which means that these pulses were short, but they were spectrally pretty narrow band pulses. And what I mean by that is this is now my nanodisk, and I'm zooming into the actual protein structure. And so in transparent gray helices, you see these protein scaffolds. And then within the protein scaffold, there are a bunch of pigments, chlorophyll shown in green, and also accessory light harvesting pigments, carotenoids shown in pink. If you take the absorption spectrum of this protein, it looks like this. Here, the rainbow bar, I'm just showing the color of each of the wavelengths. And so you can really see that the absorption actually spans across the visible from the red region to the yellow region, to the green, to the blue region of the solar spectrum, which means that we really need a broadband source to try to probe all of this energy state. But the laser that these people have been using for such a long time is really basically narrowband encompassing something like 20% of the absorption spectrum. So that was a huge challenge, which I overcame by developing an ultra broadband light source. So this is, I'm covering my spectrum, which is an ultra broadband spectrum, to sort of show you how much I expanded my spectral bandwidth. Here is my spectrum. So it's shown in the rainbow shade is what I developed. So that has a 200 nanometer fold half max compared to the 50 nanometers before. And I also implemented a chopping scheme to increase detection sensitivity by 15-fold. And so we were able to measure the scattering protein or nanodisk over the across the entire visible spectrum. And so basically I get something like this. So now we can go back to the science question. What are the ultrafast photophysics that are responsible for the dissipated fluorescence quenching that I showed you earlier? And so if you think about the scientific question, it really comes down to the interaction between these pigments or pigment electronic states, which are the chlorophyll and carotenoid. I didn't mention it to you. I didn't mention to you what electronic states contribute to each absorption region. Carotenoids, accessory pigments shown in pink, absorb the blue-green region of the solar spectrum. And then chlorophyll are really poor absorber in the green, and so that's why carotenoids complement the absorption, but they are pretty good absorber in the red, low energy, and the blue side of the solar energy spectrum. And so what that means is, okay, we know that chlorophyll has a lowest lying excited state we call Q. That is a state that emits fluorescence. And so one of the proposed mechanisms in the literature, this is one of the many mechanisms that I'll be focusing on today, but the other thing that I didn't mention is carotenoid actually has a lowest lying excited state that's actually forbidden or dark, optically dark. That is, of course, it's dark, and so nobody knows where it is, but it's proposed to be pretty near the energy state of the fluorescence state. And so if we can imagine this, for this proposal, sort of a dissipated chlorophyll Q to carotenoid S1 energy transfer, then what can happen is, it can hijack some of the excited population that was going to sit around here. And then this pathway is super fast compared to this nanosecond pathway. So we can rapidly sort of dissipate harmful energy as heat. So that was the crux of the proposal that was put forward by a lot of these other people. But then the problem is that, that hadn't been ever observed experimentally. So basically, we wanted to know, okay, is this pathway actually a thing in the system? And if so, can we characterize a timescale? Because nobody knows if it actually exists or what the timescale of this pathway is. And so what the experiment that I designed using this technique is to pump into or look at the part where I'm pumping into the chlorophyll Q state, which would be the pump energy right around here, and then look for the signal. Of course, I can't look at this signal directly because this transition is forbidden, so it doesn't show up on the spectrum. But luckily, there is another pathway, which is the excited state absorption. I'm going up from this dark state that is super strongly allowed. And so we can look at this allowed transition by looking at this pump energy. And so we know that from literature, this shows up around here. And so what I did was to look at this little corner of my ultra broadband spectrum. Here's a zoom in of that region, just plotted with a different color map at about 500 femtoseconds after photo excitation. Here I am giving you the guide, the pump energy of chlorophyll Q that we were pumping before. Here's the diagonal position of chlorophyll Q. So that's my donor, if you remember my cartoon diagram. And then we will be looking at this cross peak pumping in the into chlorophyll Q and probing carotenoid S1 and SN transition. And what I did was to compare these three conditions. So this top condition, which is labeled detergent, is the isolated non-native environment that a lot of people study. So that's our control to benchmark the photophysics. And then what I compare that to this was to this nanodisk embedding single light harvesting complex 2 protein, so I could study the role of the lipid membrane. If the membrane is there, does it actually change the photophysics or timescales? And then what I also did was to compare this nanodisk with a single protein to the same nanodisk, but with an array of 4 proteins, so I could also study the protein-protein interaction or the impact of protein-protein interaction. And so I'm going to show you first the benchmark data that we observed or took on detergent protein. And again, I'm going to focus on these 2 peaks where my donor diagonal peak, if energy transfer that we propose is happening, should be decaying relatively quickly. And at the same time, I'm growing in, population should be growing in at this cross peak. So you can actually see if you monitor this donor diagonal peak, there is a rapid decay of about 300 femtosecond time scale that shows up on our kinetic trace. And again, if you look at this donor acceptor cross peak, which is now here, I do see growth of the population, as you can see by this curvature, with about the same time scale. And so we assigned that as this proposed dissipative energy transfer based on the spectral position of the peaks and the kinetic traces. Now what we did was to, again, compare the traces between the detergent non-native sample versus the lipid membrane sample, nanodisk. And you can see that the qualitative shape of both traces are similar, but then the curvature changes. And so upon fitting, we were able to extract this 14% increase in the amplitude or efficiency of this pathway, which means that the presence of lipid membrane, which are totally neglected before in other people's studies, is actually doing causing a non-negligible change in the kinetics and amplitude of this photoprotective pathway. Finally, I'm showing overlaying the data from the array of LHCII, and you see that again the qualitative traces are the same. You know, donor decays really rapidly, acceptor grows in pretty rapidly too. And when we analyzed, when we quantitatively analyze the amplitude in a time scale, we do see another 15% increase in the amplitude of these this pathway, which tells me that protein-protein interaction, this like squishing of multiple proteins into an array, actually also does play a role in governing these photophysics. And so to wrap up this part of the story, the conclusions that we actually gained by combining this new innovative technique with a really large spectral bandwidth and sensitivity and this new biological platform was to discover that we discovered previously hidden rapid dissipative pathway that might be responsible for photoprotective mechanisms under high intensity of sunlight. And also the other thing that we gained, which was kind of not ever explored before by other people, is that we were able to, by using this biological platform, extract out the role of protein-lipid interactions and protein-protein interactions in these light harvesting complexes that might be impacting pigment photophysical pathways that might also be relevant for photoprotection. And so going back to my sort of outline slide, this second half of the story will focus a little bit more on controlling the energy flow. And I hope I am doing good on time still. Yeah, so here the question was, can we actually manipulate or control the energy landscape of these synthetic nanomaterials towards improved energy flow? This is a little hard to achieve for lateral systems like leaves because they're just like wired by nature in a certain way structurally, but these synthetic nanomaterials we can manipulate better. And so we try to sort of think about this question, can we actually make the energy flow in these systems better towards better devices and better device performances? And the platform that I utilize for this part of the story is a little photonic, and so maybe this might get you more excited, which is called excitation polaritons. So this is a buzzword. I don't know how many people have heard of this before, but this is a hybrid light matter state that is really exciting a lot of people in the physical chemistry community right now. The construction of the sample is the following. So here are 2 mirrors. These are gold. But if I can make this with other metal, silver, so these are the same mirrors as you have in your bathroom. I put a pair of these mirrors and I can trap some light, right, which will bounce back and forth because these are both reflective substrates. And so I can define some sort of energy or wavelength of the light that I'm trapping in the system. Now, I can pick a molecular or material system that has the same energy as a light. right? That has a peak right around there. And so these people have been putting out some really awesome work using this platform called Polaritons, including myself, we just published our review paper from my group for the first time in my group. But anyway, what happens when they interact really strongly because they're resonant energetically is that they split into new energy levels, and they're mixtures of light and matter because we are strongly interacting light states and matter states. And so we're going to call this upper energy level state upper polariton, or UP in short, and then this other state that's lower in energy is called lower polariton, LP or LP. In the collective coupling regime, which is meaning that you couple many different molecules. So we can couple single molecules, so just one molecule to a photonic cavity, in which case we don't get any dark states. But in the collective coupling regime, where there are many, many molecules simultaneously coupling to the same photonic mode, we do get some dark states appearing right in the middle between UP and LP. And the splitting energy is called the Rabi splitting, and this is totally tunable depending on the transition dipole of my molecule that I'm putting here, or the concentration. If I up the concentration of the molecules that I'm putting here, we get more splitting, and orientation of the, E field of the light that's trapped here, and transition dipole moment. So there's a lot of parameters that we can systematically control in the lab setting to try and control the Rabi splitting between the UP and LP. And so here I utilize semiconducting single-walled carbon nanotubes, which are diagrammatically shown here. And these are actually excellent materials for creating strong light matter coupled exciton polaritons. Because light matter coupling strength or Rabi splitting strength also, like I said, depends on the transition dipole. They absorb really, really strongly in the near IR, as shown by this peak labeled S11. So this is for molecular system, the equivalent of S1 state, the ground to the first excited state. They're really sharp and really narrow and really intense. And so we are utilizing this band to couple to the cavity. And the other thing that's kind of cool about nanotubes is simply by changing the diameter of the nanotube, we can totally control the optical property of these nanotubes. So you can kind of see how I'm changing the diameter in this plot, and you are seeing a shift in the absorbance of these peaks. And so that's another really cool feature about using these versatile platform. So here is really what motivated this part of the story, which was my collaborator, Mike Arnold, who is the corresponding author on this paper, is an expert, and he's really interested in making devices out of these carbon nanotubes, including like other organic materials, because they're good at, absorbing charges and energies and transferring energy and creating charges, I guess, in the end. But the problem is that, if you know about this, organic materials are actually terrible at actually diffusing energy around to the interface between donor and receptor. And so they get, basically, they're done here. And by which I'm calling the diffusion length of energy, which is about sub 10 nanometer, which is very, very low compared to the actual distance that it has to travel ideally to reach the interface between the donor and receptor. And so if you measure the efficiency of this schematic device, it's just 1%, which is very, very low. And so our idea can we utilize this new platform, Polariton? We can put this device into a cavity, polariton cavity, and then maybe, because we know that at least light, this orange stuff is delocalized across the length of the entire cavity, maybe you can utilize this characteristic of light to maybe achieve longer range energy transfer. And for this particular system, the cavity length here to here is about 300 nanometers. So maybe if we put this device into the cavity, can we maybe see energy transfer over a 300 nanometer distance versus 10 nanometer like shown here? So that was our dream. And I'm going to convince you that, we found some signature that alludes to the fact that maybe we see that. And so before we talk about other things, here are some of the really fundamental parameters that we wanted to study, which are what are the time scales of energy transfer if we put this thing into the cavity, and what are obviously the photophysical mechanisms of energy relaxation if we put this entire thing into the cavity. Of course, we didn't want to start with something so complicated, so we actually dumbed it down a little bit by just removing the acceptor. And so here is just a thin film layer of just carbon nanotubes. So energy should still be transferring because there's a bunch of carbon nanotubes. And so this is what we made. And in reality, what I made is a little bit more complicated than this. I don't have C60 still, but this is what I made, which is a tri-layer system embedded within the cavity, where if I zoom in onto the structure, it's actually two different types of carbon nanotubes. So I'm going to call this 6,5 and this 7,5. These are two different carbon nanotubes just based off of how I'm rolling my graphene sheet into different diameters. And then what's notable also is in the middle, I put an insulating barrier between these two layers. So this barrier is thick enough, it's 150 nanometers, and it's thick enough such that there shouldn't be any energy transfer between 6,5 and 7,5 due to direct contact. So this is basically literally acting as a barrier for direct energy transfer exchange between the two layers, okay? So the experiment that I did was, or envisioned was, okay, if we pump into the donor state and if we actually get some light out on the acceptor side in the presence of this insulator barrier, that probably means that polariton is actually doing something to govern or mediate this long range energy transfer over at least 150 nanometers. So that was the idea, and here I'm showing you some data. We're starting from something steady state, and so here I'm showing you the energy level diagram or the absorption of the carbon nanotubes. And so as you can see, 6,5 absorbs slightly at a higher energy than 7,5 here and here. And so you can see that indeed this is a donor because this is a higher lying state compared to 7,5. Now, when I put the system into the cavity, this is what you see. So you see a bunch of different things happening, but what's really notable is that the peaks are now shifting as a function of angle, right? If we change the angle of incidence from 20 degrees to 30 to 40 to 60 to 70, we are seeing the peak positions of these carbon nanotubes actually shifting, which is a hallmark of polariton formation. So for example, if I actually take a cut at this 40-degree angle, and then if I overlay that, you see that the peaks are gone. And indeed, we see a really beautiful splitting into upper and lower polariton for the system. And if I characterize or quantify the Rabi splitting based off of the peak positions, I get about 100 MeV, which is pretty large for organic materials. So how do we actually model the new electronic structure of the system in the presence of light matter coupling? So here I'm doing a very, very simple construction of Hamiltonian by on the diagonal putting in my light mode and carbon nanotubes. So there are a bunch of different carbon nanotubes. So it's not a 2x2 matrix, but this is the idea. And then off the diagonal, which corresponds to the coupling strength between these two elements, I put a G, which corresponds to my light matter coupling, okay? And then I diagonalize my Hamiltonian, and E, which I'm plotting in thick black lines, actually show me the polariton energy. And you can see an excellent agreement between the thick black lines and my experimental spectrum, which is shown in 2D contour. And so we can get out the energy levels as a function of angle. And the angle dependence is because, like I said before, we're mixing light and matter. Light depends on angle of incidence. And so that is why this means that polariton has been formed. And then I can also get out what's called mixing coefficients, which tells me, okay, how much of this state is light character and carbon nanotube character? How much of this state is light character, carbon nanotube character, okay? And so here I'm going to focus on, there are 6 states total, as you see from the number of the thick lines. But here I'm going to focus on 2 states, which are the key states in our experiment, which is the upper polariton, which is highlighting purple, which is about half light, half 6,5 donor. And then this guy is lower polariton, which is 30% light, 40% 7,5 acceptor state. So going back to my schematic diagram, I'm explaining the experiment. What I wanted to see was, okay, if I pump my upper polariton state using 2D, if I see a cross peak happening between UP and LP on my spectrum, that probably means that my energy transfer that I'm proposing is actually taking place. And so here is a 2D spectrum that I'm showing at a pretty early time, t equals 100 femtoseconds. You see a bunch of diagonal peaks appearing along the diagonal that correspond to my UP and LP energies based off of the steady state spectrum. Now I move to a later time delay, and which is 500 femtosecond. You actually see that the UP population decay really quickly, so we don't see a lot of intensity here. But then there is this cross peak that's growing in at the intersection of UP and LP, like this. And so here I'm plotting the kinetics. And like I said before, the UP peak up there is decaying super quickly within the first 1,000 femtosecond. If I now plot the cross peak intensity, it grows in on about 200 femtosecond time scale, which corresponds to the decay time scale of the UP. And so we assign this to the energy transfer across the barrier between 6,5 and 7,5, which would never happen outside the cavity. And this energy transfer time scale was even 5 times faster in thin bare film blends when they're directly touching each other. So that was really remarkable that we were seeing such a fast long range energy transfer across this thick insulating barrier. So something that we were kind of like really wondering, this was very exciting for us, but something that didn't really make sense for us was, okay, if I actually estimate the lifetime of this entire thing, this is it has a bunch of light character, and we know the light is super fast compared to matter or carbon nanotubes. So if we can actually, estimate the lifetime of the polariton using completely classical optical parameters. And so this is a number that I get out. This is 10 femtosecond. And if I plot that 10 femtosecond onto this plot, it's about this much. It's right here. So then my question is, okay, why am I seeing all this stuff happening past the first 10 femtosecond when we are estimating this entire lifetime for this entire thing to be just 10 femtosecond? And so that was our conundrum. And so that's when we turned to some theoretical modeling of this data. And so the idea behind this modeling is that we took the diagonal slice of our 2D spectrum, which I'm showing in gray line, and then that gives me basically a pseudo absorption spectrum in the steady state, as you see on the right. And then what I did was to calculate or simulate the spectrum by using my Hamiltonian that I constructed before by varying G, basically fitting for the variable G, which is light matter coupling. And this is our best fit. And I get my best fit when G is about 43 MeV. But then there's this other parameter that I had to put in, which is kind of important for this story, which is the coupling between neighboring carbon nanotubes within the same layer. Again, there's no communication between this layer and this layer. So there's no J between 6.5 and 7.5. But we had to put in the coupling between 7.5 and 7.5 and 6.5 and 6.5, which makes sense because these are aggregates of carbon nanotubes within each layer. Okay, so that's about 10 MeV, which we took from literature, actually. We didn't vary this value. We just took it from literature. We put it in, and all of a sudden, we got this perfect match between the spectrum that's calculated and measured. And then we can also plot the energy states that's covered under this envelope. And we see this really dynamic looking structure with different color codes. And so what I did was to plot the percentage of the relative percentage of the light character, 6.5 character, 7.5 character in different colors. And so you can see that the UP, some of these states are purely 6.5 like characters. There's no orange here, which means that they just behave like carbon nanotubes. But then some of these states that you see across the spectral range has some light character. So basically what I'm trying to say is that there are a lot of states with varying light character, varying carbon nanotube character, varying oscillator strength. It's a whole mess, which we call the energy manifold, created by the interplay between J and G. And you'll see why this is an interplay created by both of these parameters later. Yes.
The assumption that it's thermal or vibrational transfer within each population of carbon nanotubes, or is it also photonic coupling?
Both.
Okay.
Yeah. Yeah, so what I did now was to zoom in onto this UP branch only because that's our donor state that funnels the energy to the other states. And so as you can see, again, there are a couple states that have a lot of light character, but then there are other states that are really carbon nanotube-like and basically no light character. And so I can't do this experiment in reality because I don't have, high enough resolution on my laser. But theoretically, I can just pump design an experiment, a simulated experiment where I'm just pumping these states with a lot of light character. And so this is my measured cross peak. This is not simulated. I can simulate the kinetics under this condition. Imagine that I'm coming into super narrow band pump, and so I'm only pumping this half of the UP. And what I get when I simulate it is this. And so this is actually totally not a match with my measured spectrum, but this is also exciting because it actually reproduces the expected 10 femtosecond fast decay that's estimated from my lifetime measurement, okay? Now, I'm going to the other half where there are less light character, more carbon nanotube character. When I now simulate this scenario, I do get a perfect match between my measured and calculated spectrum, which again tells us that this manifold of varying light character and varying carbon nanotube character actually gives rise to this kind of behavior that we're seeing, the long-lived and slow sort of dynamics that I see in my experimental 2D spectrum. Okay, so basically what we concluded was that the experimental data was only able to reproduce by balancing out the light character and the molecular character. Having too much light character actually doesn't help you because you end up with this situation where nothing useful can happen. But because we were balancing out the light character and the carbon-negative character, do we see such interesting phenomena, which is this long-range energy transfer in the system. So here I'm showing you a couple more plots before I end my talk, which is going back to this modeling. But then here I totally killed my inner carbon nanotube coupling. So J equals 0 now, only light matter coupling. Again, I see a really fast spike or decay of the system that's more consistent with the estimated 10 femtosecond lifetime. It does not reproduce at all my data. And the energy states get pretty simple. There's a single UP, there's a single middle polariton, single LP, everything's generated. There's just 5 states, very clean looking, very simple spectrum. This is a picture that a lot of people had been using before we published this paper. But we just proved that this is not entirely not true. This is the real picture when we actually have an interplay between my J, which is about the same order of magnitude with G. So the matter coupling and light matter coupling have to sort of act in concert to actually realize this really interesting phenomenon on a slower time scale, which is all realized by this manifold of states. Some of these are dark in gray, right? So there's a lot of dark states, there's a lot of bright states, a lot of states with light character, a lot of states with totally no light character. This is a whole mess, but this manifold actually creates this interesting dynamics. That was our conclusion. And so basically, this was our new insight that we gained into polariton photophysics. In reality, I'm pumping everything pretty much with a broadband pump, right? Because I have such a large spectral bandwidth, I can't selectively pump any of these states. And so what I'm doing is I'm pumping this entire UP branch, and what we are seeing on our data, we think, are these superposition of all of these potential pathways, some of which are, I believe, are super fast, some of which are slower, and so the combination of all of these arrows are mapping onto our 2D spectra, which is appearing as a slow component of about 200 femtoseconds. And so this is the end of my second story, where we saw a polariton-mediated long-range energy transfer in carbon nanotubes that had not been observed before, which is very exciting. And we could actually utilize maybe this polariton platform as a way to increase light harvesting efficiency or device performance, regardless of the device architecture, because the light mode is already delocalized across the entire length of the cavity, whatever the length is. And so here are a couple slides before I really have to end. That's happening in my lab right now at BU. So I, talked about these light harvesting systems, and I really got interested in how can we actually engineer. These are natural systems, so that these are terribly hard to engineer, but can we actually control or develop some ideas to control some of the pathways toward better energy transfer? And so what I did, the idea that I came up with is to combine the both sides of my story. Okay, can we actually put these guys into the polariton cavities? Because then we can control the Rabi splitting, we can control the energy landscape, we can basically rewrite the energy landscape of these natural systems, which is otherwise super hard, using photonic modes in polaritons. And so this actually has been done, or proof of principle has been shown by a couple groups. So this is a paper from David Litzy in the UK, showing that they can keep these bacteria alive within a cavity, which is pretty cool. So this is not a spectroscopy paper, but this is a spectroscopy paper utilizing ultrafast lasers coming out of Tony Polaritons group at Lund in Sweden, where they actually put in these purple bacterial protein into the cavity, and they do see a very pronounced difference between outside the cavity and in-cavity kinetics. And so inspired by this work, we put in chlorophyll, which are the primary light harvesting pigments in photosynthesis. I didn't want to start with something as big as a protein, and so we just took the pigments out of the protein and we just put it in like this. And here's our photo of a nice thin film layer of the chlorophyll. And we can characterize the height using AFM or ellipsometry to design, make sure that we are hitting the resonance condition required by the strong Rabi splitting condition. And so we do get this nice looking smooth film of on 180 nanometer thickness. And if I now measure the reflectance or absorbance, this is the absorbance of the bare molecule shown here in gray, just flipped upside down. Now, if I measure the polariton, I do see a really pronounced, angular dependence. I'm going from 20 degrees to 70 degrees, and also with Rabi splitting between my UP and LP, which do show that polariton is being formed using these chlorophyll layers built embedded into a silver cavity. And so as we speak right now, before I had to come here, they were measuring a pump probe on this data, sample. So I don't have any data to show you other than this, but that's something that is happening right now. This is, I think, the last slide. This is a diagram of what we have in my lab right now. This is an ultra-fast, ultra-broadband pump probe. So I'm not doing 2D now, but we implemented white light generation using sapphire crystals on both pump and probe. And we're pumping our white light generation with sapphiros from my conversion, 100 kilohertz of very fast laser compared to TISAS. And they're super stable. So we were actually able to get really, really broad looking spectrum all the way from the UV to near IR. And we were able to compress using chirp mirrors this entire thing to sub 20 femtoseconds. And so that's really all I have. And I want to thank all of these people and my current group members for all the work they are doing to, allow me to give such a talk like this. And of course, thank you for your attention. And I'll be happy to take any questions. Thank you. Yeah.
Do I understand correctly that these experiments are being done, not in vivo, but in your LHC?
Right.
Is there any chance of actually doing them in vivo, or is the problem the levels of energy that are required to make measurements?
So, I mean, there's a person who actually tried in vivo experiments with the pump probe, so it's possible, but one practical challenge is that it scatters a lot because, your object size is so large. Like they actually literally put in a leaf, like in their, they shoot laser like on a leaf, and they are getting data, but because it's scattering so much, it's very hard to collect high quality data. So there's that's one thing. And then the other thing is, physically speaking, you have, so each of these proteins, the single protein that I showed you, like with little blobs, each of these proteins bind 50 molecules. Okay. And then in vivo, think about how many proteins with 50 molecules are there, right? It's going to be something like millions of molecules. When you have that many molecules, when you hit them with laser, you're going to create so many excitons or so many excited states, they can collide into each other, which is called exciton annihilation. And that creates a whole bunch of other complexity mapping onto your data. And so that is why a lot of people haven't been trying in vivo, because they can't interpret the data.
But do I understand correctly that basically you're trying to simulate the lipid layers?
Right.
Are simulating the cellular walls? Is that
not walls, but the membrane structure that are in native membranes in the plants. Yeah, but that is a correct idea. We were trying to reproduce the native environment, but in a controlled way without the artifact or whatever. Yeah.
So the broadband source, why do you need the broadband source for the polariton states? I would assume that you're responsive to very narrow wavelengths.
No, actually, that's not true, because when you have a Rabi splitting, your spacing between the upper and lower polariton can be very far away, right? Like your lower polariton can be in the IR, while at the same time your upper polariton might be in the UV. In the case of really large Rabi splitting, and so you still need a broadband to be able to map out both sides of the spectrum. Yeah. Yeah.
Getting a potential 30% increase.
Yeah.
Can you trace through how that might happen?
Could you repeat the question for the people?
Yeah. So the question was, I guess you're asking, so what can I say about, based on my results, the 30%?
What would be what's the projected route to get the improvement?
Yeah, so yeah, maybe I wasn't super clear about it, but basically what I was trying to allude to is, like, this competition between the yellow arrow, which is the light harvesting pathway, and then the gray arrow. So the gray is the energy loss mechanism here, right? Whatever goes to gray doesn't contribute to the yield of agriculture because it doesn't do photosynthesis, right? And so what we're trying to do in our field, and the biologists in the field, other fields, is this community, is to try to first understand the mechanism like we're doing, but then try to suppress that pathway so we can direct more energy to the yellow so it can do more photosynthesis, which will then increase the yield of crop and biomass and whatnot. But of course, I do not know the details of how that would be because I'm not a biologist, right? But that would be the big picture idea, if that.
But some interrogation of the with like the plants. You're some sort of special engineered light source on the plants to make this happen.
I don't think we need a light source because, I mean, there are people who are replicating the native light intensity levels, like using like different lamps or whatever in vitro, but like, I don't think that's
But what would be the input? Yes.
what would be the input.
Yeah, the output, the higher energy production.
I think maybe, I mean, one idea that I have is I think we're gonna have to mutate out some of these proteins if we can. So I talked about timescales, assuming that dissipation happens in this particular protein, but obviously this is one of the many proteins that are present in the system. So there are other proteins that are similar enough, but not quite this protein, that actually are proposed to be the site of dissipation. And so if we can knock out the protein, so these are some of the experiments that are going on right now, then like, okay, like now we're suppressing this pathway because this protein doesn't exist. And so then we have like better yield of, they're not looking at the final yield of crop, obviously, because this is all in vitro, but they are able to characterize, okay, we do see something that we can call, okay, increased yield of photosynthesis. I think that's probably one of the first steps. Yeah.
This may be a more biological question.
Yeah.
Oriented term, but how much variation is there within the plant, the whole range of plant types of plants
Yeah.
in the process and in the efficiency, you know, of the process of extracting energy from the sun or whatever.
Sorry. How much variation spectrally?
In the sense of the efficiency of the process.
Yeah.
Among, are there some that are very efficient? Are there some that are not efficient at all?
Yeah, so there definitely are some variations. I would say, you know, even if you're looking at like whether you're looking at pea or like spinach or whatever, like as long as they're plants, they're similar enough because these proteins within the same species, or I mean, I really do not know what to call it, maybe kingdom or whatever, you know, like what I'm talking about, they are homologous, which means that the proteins are minorly different. But if you now compare algae, which are, you know, not plants, but they still do photosynthesis, they're different because I think this is majorly driven by the ecological needs, right? Because algae actually live underwater, and so they need more pigments that are they're gonna thrive like in deep seawater versus like plants are exposed to, you know, the sunlight, and so they sort of like evolve like different sort of pigments, and so that then corresponds to differences of spectra. And so obviously if you have different pigments, that there are different electronic structure, and so then your pathway obviously changes. And so some are definitely less efficient or more, but I don't think there's I don't think I've ever seen a photosynthetic organisms that is terrible at light harvesting. Yeah. Yeah. Yes.
How does the 10 nanosecond time delay that you sort of seems to be the mean of the transition states compared to typical atomic transitions? Is this a slow process on the scale of atomic transitions? Is this fast? I have no characteristic scale for
Atomic transitions?
well, like is that 10 nanosecond scale you're talking about
Uh-huh.
kind of low or fast?
Yeah, 10, yeah. Anyway, I think it's still fast compared to atomic transitions because these are molecules, right? I mean, carbon nanotubes are not molecules, but we're talking about molecules in general. And I think atomic dynamics occur in the attosecond range. So they're orders of magnitude faster is my belief.
So there's a elevated state for a while and it's sort of holding on to the energy and then transitioning.
I mean, we chemists call this super fast because, you know, this is faster than most lasers that you can, you know, use. But compared to atomic transitions, obviously this is really slow. Compared to things like, you know, photochemistry, this is super fast because photochemistry happens on a minute time scale, right? And so I think it depends on like what your axes are. Yeah, but I think if you are in the atomic transition thinking, then you're right that like it holds on, it just sort of sticks around for a while and then it, you know, relaxes down.
Is it long enough to participate chemically, like in this elevated state? Is it regulating the chemical pathways?
So 10 femtosecond is not like, you know, long enough to do anything useful. So that's why there's this field in synthetic chemistry. These people don't know a lot about the physical mechanism behind it, but they're trying to design molecules with the longer excited state lifetime. But that's actually pretty easy for these they work with these catalysts that are transition metals, and they typically have large spin orbit coupling because there's metals, and so they have triplet states, which are usually longer lived because they're forbidden, right, from the singlet ground state. And so those are like microsecond timescales, so that's pretty useful because like they are way longer than the 10 femto that I'm talking about, right? Because microseconds are 10 to the -6, 1 femto is 10 to the -15, so that's way longer, but then still a little bit short for like chemical reactions because they're even slower. And so one of the directions that are happening a lot in synthetic chemistries to design molecule by putting in, attaching functional groups to lengthen the lifetime, so that they can do chemistry. Yeah.
Femtosecond's a function of just how tiny your cavity is made, basically. So if you just made a wider cavity, basically, so the light just takes longer round trip. Like, does that have any impact? Is it purely post duration effect, or does having more round trips impact anything, or is it purely just post energy driven?
No, it's I think that'll have a minor impact. Yeah, this is a cavity just like the laser cavity. Like physical size, it can be as big as they are round cavities. So I plotted them as square cavities, but they're actually round because we make them on cover slips, and these are 15 millimeter in diameter laterally. But then the thickness, which is what matters, right? The lateral dimension doesn't matter. We can make it like 1 meter by 1 meter, like who cares, right? But the thickness is about 320 nanometers. That determines the resonance between the carbon nanotube state and the photonic mode. What changes the lifetime? I think the most major factor is the thickness of the metal, because if you have a 10 nanometer silver, for example, versus like 100 nanometer silver, 100 nanometer is going to be way more reflective, right? So the 10 nanometer, just like when you're thinking about like laser Q switching or whatever, if you have a lossy mirror, that creates faster damping, which means your polariton escapes more fast, right? And so that corresponds to shorter lifetime. And so if I use actually 10 and 60 for this experiment, because unfortunately the way this experiment works, 2D works, is the sample has to absorb a lot of light, and so it can't be totally reflective, which means I have to have at least one lossy mirror. But if I were to change it to 60-60 on both sides, I'm sure that it'll live way longer, for example. Yeah.
So I think we're at 8 o'clock, and the janitor is going to come in and be upset with us very soon. But if we want to stick around and ask more questions, I don't know what your schedule is like. The lobby out there you're welcome to stay in. I'll bring the pizza out there. But we should clean up in here. So, all right.
All right, thank you so much.
Really cool.
Thank you.